Hello everyone, and welcome to today's webinar, Membrane Proteins from Gene to Cryo-EM Structure with Thermo Fisher GeneArt and Salipro Biotech. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Michael Liss, Senior Manager R&D at Thermo Fisher Scientific, Dr. Ieva Drulyte, Cryo-EM Application Scientist at Thermo Fisher Scientific, and Dr. Jens Frauenfeld, CEO and founder of Salipro Biotech AB. Welcome. You may now begin your presentations.
So, thank you very much for the nice introduction, and thanks very much for introducing our webinar today, which will be around workflow, which we designed to attain cryo-EM structures quickly when you just start from electronic data of genes and or proteins. So the entire workflow of this procedure starts with gene synthesis. So we start at GeneArt with a protein sequence, for example, back translated into DNA and design it for optimal expression. I will explain a little bit later what that is. So then, of course, starts turning the electronic data into a tangible molecule through gene synthesis and the transfection into mammalian cells for their expression. The next step in our collaboration is then with Salipro Biotech, who is specialized on one-step reconstruction and purification of the membrane proteins from the cell pellets that we send them, screen them, and do the production analysis, of course. Finally, it ends up with Thermo Fisher again, which takes the specimen proteins, prepares them for cryo-EM, freezes them, does the data collection and the image reconstruction to end up with the structure. So the first part, gene synthesis, starts, as I said, with electronic data of a protein, optimized in silico and then put into molecules through regular oligosynthesis. These oligos are then assembled to attain full-length double-stranded DNA. This is cloned into expression vector we used in the study pcDNA 3.4. Sequence verification, and then typically we ship these genes to our customers if it's just gene synthesis, but here we go one step further. What you can see here, you may know that gene synthesis is a very competitive market, so we put a lot of emphasis on miniaturization and automation of the gene synthesis process itself, and therefore attain very high throughput capacity and very short turnaround times of our services. So that was gene synthesis. After gene synthesis, what we do and also offer to customers as a custom service is upscaling the plasmids of the expression construct, and then transfect. We use mainly XP293, ExpiCHO, but also can go into insect cells as SF9. And eventually end up with a protein purification. So this entire process allows customers to order their protein of choice purified from GeneArt. I talked about optimization, so the first step still in silico. What is gene optimization? It's a multi-parameter algorithm that takes several parameters into account that contribute to high expression of genes. And that's very obvious. It's the codon usage, which is not equal in every organism. And the GC content, you may know that high GC content genes express better in mammalian cells. Of course, we eliminate cryptic splice sites that may be present in the cDNA or in the DNA of other origin. Avoid instability sequences, secondary structures, and so on. So the list is long, it's up to 20 parameters. And that algorithm is actually not trivial because the algorithm tries to find the best possible solution given all the possible codon combinations, which is of course an astronomical high number. So does that actually work? Gene optimization, and here's some data from a paper we published about 10 years ago, in which we took 50 standard human genes and optimized them for human expression. So you may think, well, human gene, why isn't that already optimized for expression? But of course, the genes that we have in our body are not optimized for highest expression, but for highest functionality in the concerted network of the cellular metabolism. So here's the data from some data from that study, 5 sample proteins from the 5 different classes. So for example, the first one, the transcription factor CREB1, you see in triplicates the wild type expression on the left-hand side versus the optimized expression, and the same is true for ribosomal or other proteins and kinases, membrane proteins, and cytokines. So on the bottom you see the cytokine is barely visible only in the optimized version, while the wild type version gave no detectable expression at all. In total in the study we observed that from the 50 proteins, 86% of those had a significantly better expression than wild type, 10% were indistinguishable from wild type, and only one case, so 2 cases, 4%, had a lower expression than the optimized. So it's not bulletproof, but gives a pretty good result. Here's something about non-membrane proteins, what customers can expect when they order from us. This is examples from antibodies expressed in ExpiCHO, either in 24 well format or down there it's 96 well format. So you can see heavy and light chain on the top. Comasi, it's just conditioned media, so crude supernatant from the cells which you can obtain, and down there is the purified antibody, which we also offer. On the right hand side you see different QCs that we offer, size exclusion, capillary gel electrophoresis, and mass spec. So in total, what you can get from GeneArt is not just gene synthesis, but also protein expression, which is all in-house, so there's no wet work left on your side. If you choose to use this service, all the reagents are proprietary, reliable, and optimized for high expression. The scales that we offer range from 30 milliliter up to 25 liter in waveback format, but we also offer high throughput formats in 26 and 96 multi-well formats. So, coming to our actual project here, where we concentrate on proteins, we chose 4 representative proteins, 2 ion channels and 2 GPCRs, which are of clinical relevance, either in cancer, in HIV infection, in pain, and designed these to have high expression in mammalian system. We added to some of these proteins precision cleavage tags and EGFP, and also, of course, detection tags like His, flag, or C tag. Then we expressed those in 293 cells in 5-liter scale, and the washed cell pellet was then shipped to Salipro, and I will here hand over to Jens to continue what they are doing, what their magic is, what they are doing with our cell pellets.
Thanks a lot, Michael, for the introduction. Welcome everyone. My name is Jens. I will basically describe the workflow now once we receive the cell pellets from Michael and his team. Just a few words on our company. We are located here in Stockholm, and here on you can see the view of the city, and where this little dot are, that's approximately our location, so rather in the city center. We are collaborating with pharma and biotech companies and have a fully owned IP portfolio that covers our platform technology. Today, we have 38 discovery programs in several stages with 15 partners or collaborators, including 7 of the top 20 pharma companies. And here you can see a selection of some of our partners and collaborators. And what we're doing at Salipro Biotech is we're working with membrane proteins. And Michael already introduced membrane proteins to us, explaining their relevance in the drug discovery process. So starting from scratch, if you look at the surface here of the cell, you can see you have the cell membrane, and there's a couple of proteins sticking out of the membrane, which are the membrane proteins, and they're really interesting drug targets. So this one membrane protein here in the front could be a very interesting drug target in oncology. The protein in the back could be a very interesting drug target in diabetes or in pain, for example. So all in all, they're really interesting for drug discovery, but they're pretty challenging to work with. And the challenge actually starts with a membrane, and this is what you see here. So in orange, you can see the membrane protein, which is then embedded in this gray lipid bilayer. And we're moving towards a more schematic version that you can see here on the right, where the membrane protein and then the transmembrane regions. And you can see that the membrane protein is surrounded by this lipid bilayer. And those lipids, this lipid bilayer, has a very strong effect on the function and the stability of the membrane protein. So you can see, for example, those lipids, they have charged head groups. So there could be positively charged head groups, and there could be negatively charged head groups. And those head groups, they specifically interact with specific side chains of the membrane protein. Same is true for those acyl chains from the lipids that have different lengths, different angles, and they also specifically interact with the transmembrane helices of the membrane protein. And our philosophy here at Salipro Biotech is actually that the membrane protein and the lipids, they together form a unit, basically. They belong together. And if you want to investigate a membrane protein properly, you should make sure that you have the lipids around. One of the problems is in the drug discovery process, and especially for cryo-EM, at some point you want to isolate your specific membrane proteins from all the others, basically, because you want to get the 3D structure, you want to make specific antibodies. So there's several reasons why people want to isolate the specific membrane proteins out of the cell membrane context. And the most common approach to do so is to use detergents. And those detergents, what they're doing is actually they are replacing those lipids, and they make the entire membrane protein soluble to bring it into solution so you can isolate it. The problem is that those detergents are very often just a poor mimic of those lipids, because they're lacking the specific charges, the specific acyl chains, for example. And very often you lose the stability or functionality, or you have maybe also strip away some important binding partners, or you maybe disrupt the entire association of the membrane protein with its partners, basically. So detergents are a bit tricky to handle, basically. There's And there are several ways to work around that problem. And one way, for example, is that you could engineer your membrane protein, for example, by introducing thermostabilizing mutations. You could, for example, add other domains, for example, but it always involves heavy engineering, and this is a more non-natural approach, basically. And it takes a lot of time also to find that specific mutations. Another way is basically you just cut away the transmembrane part and work with the soluble regions, or just peptides, basically. But here again, this is a very unnatural approach and very artificial. You don't know if this is the real thing or if it's too much engineered, actually. Other approaches which are not entirely relevant for cryo-EM, but also being used in the drug discovery process is that you are using cells expressing the target of interest, or VLPs, but they also come with pros and cons in the process with a high background noise and batch-to-batch variations. So in general, there's a lot of really interesting membrane proteins out there, which could be really interesting drug target. But the problem is with the current approaches, very often those membrane proteins are undruggable, basically, because you can't really work with them due to this lack of stability and functionality, basically. And basically, that's when we try to find a solution for this major problem. And we basically started then back from the cell membrane. So we've developed the Salipro platform basically to solve some of those problems that I've just described. So we start with a membrane protein in the cell or in the cell membrane. And then in the next step, we're adding the Salipro scaffold protein, and which has a high affinity for lipids. And this Salipro scaffold protein would basically grab the membrane protein together with some of the lipids that are surrounding the membrane protein, and then scoop them out and then form a complex which contains the membrane protein, a number of lipids, and then the Salipro scaffold, which holds everything together in a ring-shaped format. And here in the next slide, you can see representative cryo-EM structure, which was published in Nature in 2018, where you can see an ion channel in orange, and then you can see in blue, you can see the scaffold protein, how it makes a ring around the transmembrane regions and some lipids of the membrane protein, and keeping everything in shape, and then which then enables the researchers to basically investigate the 3D structure or use it for drug discovery processes. And there's several publications in the public domain now showing the use of our technology for structure function research on specific drug targets, basically. So we have now this very bold claim by saying, well, the notion that the majority of membrane proteins is undruggable, maybe it's no longer true, because if you start using the Salipro system, then maybe that fixes some of the problems and you can actually start working with some of the more challenging membrane proteins. And as Michael already introduced, we wanted to showcase this with a couple of targets, 2 GPCRs and 2 ion channels. And we heard the introduction from Michael from GeneArt that we've chosen 4 different targets, which were overexpressed in 293 cells, basically. And then they shipped us the cell pellets, basically, and that's where we took over. So here at Salipro, we have the Salipro platform and the process which is called direct membrane extraction, DirectMX. And what we've done is we've basically, for every single of those membrane proteins, we've adjusted and tailored our platform technology for this specific drug target, so that in the end we have a stabilized membrane protein in the Salipro system that is then ready for cryo-EM, which we would then ship to Ieva for further investigation on cryo-electron microscopy. So the first target that we started working with is an ion channel called Panexine. When we received then the cell pellets, and this is basically like an overview of what we call DirectMX, or direct membrane extraction. So we have here the cell membrane with Panexine that contains a strep tag and a GFP, but there's also other membrane proteins that you can see. And in the first step, basically, we are adding the scaffold protein to the cell membrane, and then in the second step, this will then basically extract all of the membrane proteins. And this is something that we call the Salipro membrane proteome, because basically we have all membrane proteins extracted. But then the next step, we want to isolate the membrane protein of choice, in this case pannexine, and then here we used the strep tag and used affinity chromatography and then immobilized it on the matrix, and then we used we cleaved off actually the GFP strep, and then we could elute the purified pannexine, Salipro pannexine particles, whereas all the unwanted membrane proteins are in the wash or in the flow-through fractions, basically. So this is now, if you then take the elution and put it on a size exclusion chromatography, then you will get the following profile. And please note that the buffer is only herpes and salt. There's no detergent, there's no additives. It's a very plain buffer setup that we have here. And we can see here a peak that corresponds to salipropannexine. If we analyze those fractions, you can see it here. We have the band for pannexine and another protein band for the scaffold protein that you can see here. And then if we pool the specific fractions, and then what we usually do is we freeze our samples, and then we thaw them, and then we analyze them again on size exclusion to make sure they're surviving the freeze thawing, but also we're testing thermostability on defined terms. In this case, we froze it, we thawed it, and then we kept it for 16 hours at 4 degrees or for 16 hours at 22 degrees. And you can see here in the sec profile, there's no changes, even after longer incubation at elevated temperatures. So we have basically extracted this pannexine from XP293 cells directly. They eluted as pure and homogeneous particles, and they were really stable even after flash freezing, thawing, and then the subsequent incubation at different temperatures. So with this sample, then we basically then shipped and sent it to Ieva for further investigation. And now you have to be patient until it's Ieva's part to show you more about the structures. In the meantime, we continue here with a GPCR CXCR4, which was also part of this array of different drug targets that we chose. Similar purification strategy like before. I'll jump directly to the size exclusion chromatography program. So in here we can see CXCR4, the bands which are visible here, and then we see the scaffold protein bands. And here again, we then collected those fractions, we pooled them, and then here again, we then flash froze the sample, we thawed it, and then subsequently we incubated it at different temperatures. In this case, it was 24 hours at 4 degrees and 24 hours at 23 degrees. And note that for all those gene sequences, we basically chose the wild type. So there's no thermostabilizing mutations in this one. And again, the buffer is just herpes and salt. There's no additives, no ligands, just basically the protein and a very aqueous buffer. And again, the particles were homogeneous, pure and stable, even after longer incubation. And then again, we basically took this sample and sent it over to Ieva. And again, stay tuned for 3D images of this membrane protein when Ieva is going to present us more about that. Then we continued with another ion channel, TRPV3 in this case. Similar to before, we did the affinity purification, we did the size exclusion, and then we pooled the fractions, the relevant ones. And you can see, again, after incubation, after flash freezing, thawing, and then subsequent incubations for 16 hours at 4 degrees and at room temperature, that TRPV is also very stable. And the protein, the SDS page is also indicating that we only have subpocene present and then the TRPV3 bands, basically. Last but not least was then another GPCR A1R. Here again, similar setup. So we started with the affinity purification, size exclusion, and then continued with the analysis. And here again, flash freezing, followed by thawing and subsequent incubation at 4 degrees and at room temperature. And we have a pretty robust setup of A1R as well. So these were the 4 targets that we selected, and then basically shipped them to Ieva, and then from here on, Ieva then present you more about the general setup of cryo-EM, and then more specifics on the drug targets that we investigated. So thank you, and then happy to catch up later.
Thank you, Jens, and hi, everyone. I'm Ieva, an application scientist at Thermo Fisher. And let me tell you now about the last step that these proteins took in their journey from Germany to Sweden and then to the Netherlands to go on our microscopes. Okay. So let's start more generally. Drug discovery pipelines can consist of soluble proteins. It could be small, it could be antibodies or multimeric complexes, or it consists of membrane proteins such as ion channels and GPCRs. But unfortunately, up to 60% of the pipelines are not structurally enabled, and that could be because the targets are not known or they're disordered. They could be hard to purify or to crystallize. Cryo-EM does not require crystals and can deal with some degree of sample heterogeneity. So this technique is well suited for determining structures of membrane proteins. And by using cryo-EM, we can increase the percentage of pipelines that can benefit from structure-based drug design. So cryo-EM is impacting drug discovery on various types of targets. For example, cryo-EM can be used to gain insight into targeted protein degradation or formation of high-order target antibody complexes, such as rituximab and CD20. We can use this technique to structurally enable targets which resist crystallization or adopt many conformational states, for example, kinases. And cryo-EM has been successfully used to determine many structures of trip channels and high-resolution structures of GABA receptors with ligands, and also active state GPCRs. And evidence to the value of cryo-EM for membrane proteins can be illustrated with this graph over here. So historically, X-ray crystallography was used for determining the structures of ion channels, but as of 2020, cryo-EM has taken over, and as you can see from this graph, most human ion channels are now structurally solved by cryo-EM. And you can see an example of one cryo-EM structure of an ion channel. Here on the right, this is SARS-CoV-2 3A ion channel. And now let's get back to our workflow that we're presenting in this webinar. You heard from Michael about how the genes were synthesized and proteins were expressed, and from Jens about how these membrane proteins were purified. And now we'll talk about the cryo-EM part. So there are 4 major steps in this workflow. So first, the samples need to be plunge frozen to make a really thin layer of particles in vitreous ice. And then we want to screen these samples and collect data on a transmission electron microscope. And then we end up with a stack of raw images. And then in these raw images, we want to take particles, extract them, classify them, and take the best stack of particles forward to reconstruct a 3D volume. So the first step is really important in this workflow, and is cryo-EM grid optimization. All samples that I will talk about were vitrified using quantifoil R1.2, 1.3 grids, and using a Vitrobot Mark IV plunge freezer. And grid optimization involves vitrifying samples and making sure that the ice is thin enough and you have the right concentration of particles in ice, and that these particles adopt random orientations as much as possible. But it is a well-known factor in cryo-EM that particles do not always adopt random orientations, and they like to adopt preferred orientations in this vitrified ice. So to improve orientation distribution and to help with the reconstructions, just right before plunge freezing, I added a little bit of mild detergent, so fluorinated phospholine 8 below the CMC level, just to improve this orientation distribution. And you can see here for the Salipro Biotech 1, adding a bit of detergent helps to improve these orientations, and we could see a lot more side views in 2D class averages, and similar effect was observed with Salipro CXCR4 as when we added more of a detergent, we saw a better angular distribution plot in the final reconstruction. So let's now go and jump into the processing of the data for Connexin 1 channel. And in total, I collected nearly 8,000 micrographs in a one-day session on the microscope on the cryo-EM. And from this data set, I picked 1.3 million particles and performed 2D classification. You can see some representative 2D class averages on the top right. Then most of the processing was done in Relion, and this was followed by 3D classification. So you can see the 8 resulting 3D classes, and I chose the one on the very right, boxed in green, and took all the particles from that class and refined them to produce a 3.1 angstrom refinement. And in the bottom right, you can see an angular distribution plot. So now let's have a look at the cryo-EM density. You see the Connexin 1 map in yellow-orange, and when you lower the threshold, you also see there's a potent scaffold protein in transparent blue. And yeah, we could see that the channel has C7 symmetry, and yeah, most of it was really well resolved, and we're in the process of now building a model. So let's jump into the next target. So that's TRPV3. And I want to say that I received a sample from Stockholm just last Thursday, and it went under the microscope into our Glacios cryo-EM last Friday. So this is really hot off the press. This data, I made some grids and I screened them. The ice was a little bit thick, so I think this project needs a little more optimization to get thinner ice, but nevertheless, I collected a data set after the screening, 2,500 movies, from which I picked a little bit over 500,000 particles. And here on the right, you can see 2D class averages of Salipro TRPV3. And after 2D classification, I took the best 150,000 particles for ab initio reconstruction, and then the third class, the one with a green box, is the best. It shows the protein of the right shape. So I took these particles, about 100,000 particles, and performed non-uniform refinement to get a 4 angstrom reconstruction. And in this slide, now you can see the cryo-EM map of Salipro TRPV3. You can see that it consists of 4 subunits. And once again, when you lower the threshold, you can see the supposing scaffold in transparent blue. I could also fit the existing structure of TRPV3 into the density, and it corresponds really well. So now the next steps are just to do a little bit more of grid optimization and improve the resolution. Okay, and now to the final of the 3 proteins I will talk about. So the final one is the chemokine receptor, the CXCR4. Yeah, for this project, I collected 6,000 micrographs, and from these micrographs, I picked 300,000 particles and performed 2 rounds of 2D classification. So on the top right, you see 2D class averages. You can see that there is definitely different views, some side views and some top views. And then the best class particles belonging to the best 2D class averages were taken for ab initio reconstruction into 5 classes. And you can see the best ab initio class boxed in green. And this class consisted of 64,000 particles. And I extracted them and refined them to produce a 3.2 angstrom map. What's also interesting to mention that when I collected the data, I performed motion correction on the fly, and then I took the align images and uploaded them to our S3 bucket on AWS, and I did the rest of the processing on AWS A100 GPU instances. And I calculated the time that it took, the GPU time that it took to go from the motion corrected images to the final reconstruction, and it was a little bit less than 12 hours, which just shows how quick the processing can go for such a project. So now let's have a look at the cryo-EM map. You can see that CXCR4 is a tetramer, and we can see a supposing disk in transparent blue. I took a, sorry, need to change the slide. Yeah, so I took the CXCR4 model from the AlphaFold 2 database, and it fits really well in the map. And you can see on the left side that the side chain density corresponds well with the bulky side chain. And lastly, I also wanted to bring your attention to some additional densities that are visible in the EM map. So as Jens was saying, that when these proteins are purified with supposing scaffolds, lipids can be scooped out of membrane together with the protein, and we think that some of these additional densities might be exactly that. So there are definitely additional long tubular densities visible between the subunits and also on the outside of the protein, as well as in the inside when we look for the central slice. So these could be lipids or other hydrophobic molecules, and we're now in the process of investigating what these are. So this is my last slide, and I would like to pass the presentation back to Jens.
Excellent. Thanks, Ieva. Yeah, now wrapping this initial presentation up. So we've seen basically how we started from basically just genes that were synthesized and then overexpressed into crude cell pellets. Then we tailored our platform technology on each specific membrane protein, and then we sent those samples to Ieva, who then, as you could see, in a really straightforward process, generated 3D structures of the samples that we sent. Basically, the only one missing is A1R, but this is basically work in process. Looking at the structures that we have so far, so we have Panexine, there's already a couple of cryo-EM structures out there. We have TRIP-B, there's also a couple of cryo-EM structures out there. And then we have CXCR4. And for CXCR4, there's crystal structures which are engineered with like thermostabilization mutations or some inserts basically, and they come with a ligand, but and they all of those CXCR4 structures were obtained from detergent extracted membrane protein basically. So we wanted, since we saw a tetramer, we wanted to investigate a bit further basically with doing some biochemistry as a follow-up to see what we're looking at basically. So we have CXCR4 as Ieva presented as a tetrameric form, and we already heard that it's actually straightforward to fit in the AlphaFold prediction of CXCR4 into the density that we observe, and then we wanted to investigate. Okay, but does the setup that we have is it relevant? And then in this case, so we actually have a look at the buffer where we have HEPAs and salt, and we have a very stable and pure CXCR4 that you can see here, and we've seen the structure out of it. So in the next step, we wanted to investigate, well, how does an antibody, which is known to only bind to functional and structural properly folded CXCR4, how does that behave with our setup? And there's There's an antibody called 12G5, which is also showing to be efficient in mouse models, for example. So it recognizes CXCR4, which is expressed on cells, and we wanted to see, does this specific antibody do the same to the CXCR4 that we have in the Salipro system, basically. And then here we're looking at size exclusion profiles where we have Salipro CXCR4, and then we have another peak which then corresponds to this antibody. And then upon incubation of this antibody with CXCR4, we see the formation of a complex which is corresponding to Salipro CXCR4 with 12G5, which actually means that this 12G5 actually binds to the CXCR4 as we've seen, validating the structure that we've observed before, what Ieva presented. Then CXCR4 is known to bind its natural ligand, which is called CXC12. And here again, we did a similar assay. So we used Salipro CXCR4. This is here the control. And then we incubated it with CXCR12 to see if we can observe a binding. And indeed, we can. So if you look at the size exclusion profile, if you then take those fractions and analyze them on a gel, you can see that we have CXC12 present here, which co-eludes at exactly the same position where we have CXCR4 as well, basically. We wanted to investigate this further, especially with CXC12, and we used, in this case, mass photometry. We can see here that Salipro CXCR4 has a size of approximately 350 kilodalton, and then when incubating this with CXCR12, we see a shift towards 370 kilodalton. So confirming again what we saw in the SEC profile before, that yes, the CXC12 does interact with CXCR4 in such a setup. And then there's an antagonist which is called AMD 3100, and we wanted to see if this has an effect on the binding of CXC12. So when adding this antagonist, there's actually no major structural or size difference in the particles. However, then if you have this antagonist present and then you're adding CXC12, we do not observe any binding, again indicating that this antagonist is actually locking CXCR4 in a state which makes it not possible to bind CXC12. So with this then we can summarize that. So we have a human CXCR4 wild type, basically, which is reconstituted directly from a human cell line into a lipid environment, basically, using the Salipro platform. This CXCR4 is in upper state, so it has never seen any kind of ligand during the purification process. With the experiments that we've showed here, we could show that it binds the natural ligand CXC12. We could also show that it binds this 12G5 antibody. We could show that the antagonist ADM 3100 inhibits the binding of CXC12. So in summary, actually, we have a nice tetrameric structure of CXCR4, which actually is capable of binding CXC12 and 12G5. So interesting structure. And to summarize it, so we've basically now gone from genes to structures within a very straightforward setup together with Thermo Fisher, where we have Panexine, where we have CXCR4 and TRPV, and waiting for A1R, which is going to happen in the next couple of weeks. And then together we can basically highlight or showcase this process, basically where we start with an idea of a drug target, which is then translated into gene synthesis, overexpressed in cells, shipped to Salipro Biotech, where we then tailor our platform technology to each specific drug target, basically, and then having this highly purified membrane protein in the Salipro system, and then making it directly ready for structural biology with Ieva. Yeah, so with this, I think we can conclude this little webinar. We would like to thank the Thermo Fisher Scientific teams from GeneArt and the Cryo-EM team, and all the team here at Salipro Biotech, some collaborators, and then we also have a white paper already out, which you can which you can access. And yeah, we'd like to thank you for your attention, and we're happy to take questions now.
All right, thank you, Michael, Ieva, and Jens. I'm so sorry for mispronouncing your name, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question: if two proteins interact with the extracellular domain, would the nanoparticle encapsulate both proteins?
Yeah, so we had cases basically where we have a membrane protein, which is, for example, just a single transmembrane helix, and then it has a large extracellular domain, and they might interact, and then you could extract this setup basically. So the simple answer is basically whatever you have in a cell membrane, whatever arrangement you have in a cell membrane can be extracted with a Salipro system. So either it is a stable setup, and then we can extract it as it is, or you can even enhance this interaction by adding ligands or binding partners that would enforce the dimerization or oligomerization of whatever you're interested in.
All right, thank you, Jens. Our next question here. Is there a size limit for the membrane proteins that the Salipro scaffold can work with?
No, not really. I mean, we have everything from single transmembrane helices to 56 transmembrane helices. So the elegance of the system is actually it really adjusts to the size of the membrane protein. So it will be several copies of the Salipro scaffold basically, which form a ring around the transmembrane part, and the larger it is, you just have more copies actually, which then align and form a very stable, energetically stable lipid Salipro membrane protein setup basically. So it will always adjust to the right size of the membrane protein.
All right, thank you again, Jens. Let's see, next question here. Does the Salipro method extract membrane proteins from all compartments, ER, Golgi, etc., or just the plasma membrane?
No, that's any kind of membrane works fine. So it also works for E. coli membranes, insect cells. I mean, if you can separate, for example, mitochondria or ER membranes from other membranes, then this is then the starting point. And then you can extract from those subfractions basically. So whatever is a membrane is fine.
All right, great. Thank you again, Jens. The next question: what is the difference between the Salipro platform and lipid nanodiscs?
Yeah, well, I mean, essentially what I just said before regarding the size. So with the nanodiscs, this is a very long scaffold protein. So it has a defined size. And that's where the problem starts, because in order to make it stable, you have to adjust the membrane protein and the amount of lipids, basically. They have to match perfectly for the size of the scaffold. And there's now several scaffold proteins, nanodiscs, which have different sizes, but you have to screen basically for the right size. So in the end, you have to adjust your membrane protein to the size of the scaffold protein, and our system is just the opposite. So our system will adjust automatically to the size of the membrane protein. This is one factor. Another factor is this direct extraction, where you can basically start directly from the cell membrane. That's not really possible using the classical nanodisc technology, basically. So two elements in that regard.
All right, thank you for clarifying that, Jens. I appreciate it. Our next question: in the DirectMX approach, is the membrane protein directly extracted using the sapacin with no detergent used to first solubilize the membranes?
So in the very first step, because membranes, especially when they work at 4 degrees, they're pretty rigid, basically. So in the very first step, when we contact the Salipro scaffold protein with a membrane, we add a very mild detergent, but only the first step, and all subsequent steps are completely detergent-free, basically. So only initially to disrupt the membrane partially, and then everything is detergent-free.
All right, thank you again. Next question is: does Salipro only work with the protein which has large transmembrane domains, i.e. GPCR with 7 TMs, In another word, will a protein with a very small TM domain benefit from the Salipro?
Yeah. So as I said before, like I mean, the system will always adjust to the size of the membrane protein, and we also have cases where we have a single transmembrane helix, and then, for example, dimers or trimers, basically. So it will adjust and then solubilize that, and then yeah, you can visualize it or use it for whatever antibody development or SPR.
Okay, great. Thank you. Next question. Are there any size limitations of the Salipro system? For example, the radius of the TM domain. Also, is the size of the Salipro discs the same of the same protein always the same, or does it vary a lot between each particle?
No, as you see before, I mean, first of all, the size is always defined by the membrane protein, and then the scaffold will always adjust to the size. And as you've seen from those size exclusion profiles, we have a very defined distribution. So we always have the same size, and maybe Ieva can also comment on that. That's basically what you see in the cryo-EM. We have a very defined particle sizes there. Otherwise, you wouldn't get to high resolution if the particles were of different sizes.
Are you agreeing there, Ieva?
Yes, no, exactly. So if the supposing skirt was different sizes, then we would not see it so defined when we lowered the threshold. And I showed it for each of the 3 proteins. So it's consistent.
All right, thank you both. Next question here. What concentration of membrane protein is ideal for cryo-EM, and what is the sort of minimum acceptable concentration?
I think ideally you want to have 5 to 10 mg per ml. Anything less than that, then you might have to collect a lot of data. So for all of the projects that I spoke today, I used, so actually for connection 1, I only used 2.5 mg per ml, and I've collected a little bit more data, and for the other 2 projects, I used about 5 mg per ml. So that's for ion channels, and for really small GPCRs, from my personal experience, especially just receptor-only GPCRs, the higher concentration, the better. So to form really nice monolayers, you want to have 10 or more mg per ml for small GPCRs.
Thank you, Ieva. Next question. Let's see. For the cryo-EM, what is the average size of the Salipro disc? Salipro plus lipids included. Are you able to see also the Salipro during the reconstruction? Would you say that the Salipro belt is more tight than, for example, nanodiscs?
I think the question about the molecular weight would go to Jens, because you've done more size exclusion chromatography, so you see the increase in size. Could you clarify that?
Yeah, I mean, we see that sort of, I mean, we see, for example, I mean, if we can actually say that we have, I mean, with CXCR4, for example, we saw that it's a tetramer, but the actual size was approximately 350 kilodalton, so we have 4 times the GPCR, and then the rest comes from lipids and the Salipro scaffold, and I think we have approximately, I'm not entirely sure, 8 or 10 Salipro scaffold proteins which form the ring around that one. But again, the size is always defined by the membrane protein plus the lipids which surround the membrane protein, and then Salipro just sticks to the lipids essentially. So
what you're saying is that for CXC it added like 100 plus kilodaltons, right, to the mass.
Mm-hmm.
And then other questions. Can I see the Salipro during the reconstruction? Yes, so I can see it in the 2D class averages and in the 3D reconstruction when I lower the threshold, and I can't really compare it to nanodiscs directly because I have a limited experience with nanodiscs. And yeah.
All right, thanks, Ieva and Jens here for helping us out with that question. Our next question, the CXCR4 example, the images show CXCR4 forms a tetramer. Any idea that this is physiologically relevant?
That's why we also did these experiments with the chemokine and the 12G5 antibody and the AMD 3100 antagonist. So with the experiment that we did, we feel that and also the good fit from the previous CRISPR structures that were solved. So we think this is physiologically relevant. We've seen basically in literature that there's dimers or monomers of CXCR4. There's also tetramers have been reported in some cellular assays. I mean, I can just say we used a very gentle approach. So we used the human cell line, we used the human gene, we used the wild type. We didn't add any ligands basically, and then we used the softest extraction possible. And then the result was actually we got a nice structure, and it was capable of binding this 12G5, the chemokine, and this antagonist basically. So with that, we would say yes, but no.
Okay, thank you, Jens. Our next question is for Ieva. How are the particles picked? 2D and 3D classes selected automatically or manually?
Everything was selected manually, just by examining by eye and picking classes that look sensible without deleting too much data too early on.
Thank you. Okay, next question. Let's see. How are you planning to improve the resolution for SBDD?
So, of course, I think there is a resolution limit for proteins because they might be just intrinsically flexible to a certain degree. But for example, for the CXCR4, the data that I showed today was collected actually from one of the first grids that I made without too much optimization. It was quite difficult to find good areas on the grid, and I also collected it on a microscope without the energy filter or cold seg. And I'm basically planning to do a little bit of grid optimization and use a better technology and see if I can improve the resolution.
All right, thank you, Ieva. We have time for a few more questions. This question is for Jens. Due to the flexibility of Salipro nanoparticles to adapt different membrane protein sizes, have you observed any co-extraction of contaminant membrane proteins that are physically close to the protein of interest within the membrane?
Not really. I mean, we can just say basically whatever you have in the membrane, basically, you will get extracted and then incorporated into the Salipro system. And if it really binds to a membrane product and it sticks during the purification process, it may not be a contaminant, but maybe like some physiological relevant protein that just sticks to its membrane protein, basically. So, no.
All right, thank you, Jens. Looks like we have time for one last question here. When expressing membrane protein /GPCR using ExpiCHO or 293, did you try Membrane Pro ExpiCHO also? Which one gives higher yield for recombinant membrane protein?
I can take that one. So no, we didn't try that. We went directly into ExpiCHO cells and haven't done any further comparison. So can't comment on that.
All right. Well, thank you, Michael. And thank you all, Michael, Ieva, and Jens, for your time today. Do you have any final comments for the audience?
Yeah, well, thanks everyone for really interesting questions. Please follow up with us afterwards, because I think we have received lots of questions and I think we didn't have time to answer all of those. So feel free to send us an email or get in touch with us, and then we can follow up offline or give us a call. Yeah, and would like to thank everybody from this collaboration for their contributions. I think it's really great fun and great success working with you guys. And again, thanks everyone in the audience for attending this webinar.
All righty.
Second, Jens, it was a really interesting collaboration, and I'm also excited about our last target, so a really small GPCR protein, A1R.
Yeah, agree. A lot of fun, really.
All right, great. Well, thank you again, Michael, Ieva, and Jens, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.